Hey guys, we're going to go over the protein synthesis review worksheet. So if you have your packet, it is pages 8 and 9 because it does include the flowchart. So let's go ahead and go over this just so you can get the right answers. Again, the keys are on the blog as well if you would just prefer to read the keys. Number 1, how are DNA and mRNA alike? Well, for one, they both contain genetic information. Okay, they're made up of nucleotides, and they're both nucleic acids. So those would be 3 similarities between DNA and mRNA. 2, how are DNA and mRNA different? DNA is in the shape of a double helix, whereas RNA is single-stranded. The bases in DNA, cytosine, guanine, adenine, thymine, CGAT. Bases in mRNA, cytosine, adenine, uracil, guanine, CAUG. Instead of T, you have U in RNA. Sugars for DNA are deoxyribose. Sugar for RNA is ribose. DNA is only ever in the nucleus. It's too big to leave. mRNA is basically throughout the cell, wherever it wants to be. First part, transcription. Transcription is when DNA goes to or is used to make mRNA. 1, how many strands of mRNA are transcribed from the 2 unzipped strands of DNA? 1. So we're only wanting to make that 1 mRNA strand. 2, if the following were part of a DNA chain, what mRNA bases would pair with it to transcribe the DNA code onto mRNA? Okay, what pairs with G? C. What pairs with G? C. What pairs with A? U, because we're making mRNA. I'm going to keep reading. So the first codon on our mRNA is CCU, second codon AGC, third codon GGA, fourth codon AUC, fifth codon UUA, and then you have one letter at the end, which is a G. 3, if DNA is described as a double helix, how should mRNA be described? Single-stranded. Easy. 4, how are the accuracy of DNA and mRNA codes assured? So RNA polymerase, which is what makes the mRNA, is going to have this kind of, it's like spell check for itself that you use on Microsoft Word or whenever you're typing up something and it underlines a word if it's spelled wrong. RNA polymerase kind of has the same thing. And so if they didn't have that, we would have mutations going on all the time. So mutations are pretty random and rare. They're rarer because RNA polymerase and even DNA polymerase has these proofreading mechanisms that help to prevent those mutations from occurring. And then after transcription, after you've made your mRNA, the mRNA leaves, goes to the cytoplasm, and then you have translation, which is when mRNA is used to make a protein. 5, name and describe the 3 types of RNAs. You have tRNA, which is transfer RNA. Transfer RNA contains an amino acid, and at its bottom contains an anticodon that pairs with mRNA. So the whole point of tRNA is to bring whatever amino acids we need to the ribosome. You have rRNA, which is ribosomal RNA. Basically, it just makes up a ribosome. And then lastly, you have mRNA, which is messenger RNA. This is what transfers genetic info from the nucleus, where the DNA is located, to the ribosome. So it's literally the messenger. It's carrying the information we need from the nucleus to the ribosome. 6, what is located at each end of the tRNA molecule? At one end, we have an anticodon, which are 3 bases that will pair with the codons on mRNA. And at the other end, they have that amino acid. So that's why they transport the amino acids to the ribosome. How many bases are needed to specify an mRNA codon? 3. You always read by 3 bases. I'm just going to go ahead and read. So we have a strand of mRNA. We're trying to make what the tRNA anticodons would be. Okay, I'm just going to read the answer. A, U, C, G, A, U, A, G, U, 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 A. Remember, tRNA is still a type of RNA, so you're still going to have U's. You should not have any thymine in any type of RNA. 10, how does mRNA get out of the nucleus? It leaves through the nuclear pores, which are basically just openings in the nuclear membrane. 11, what is the difference between an amino acid and a protein? Okay, so the amino acid's the monomer, protein's the polymer. So when amino acids are put together by peptide bonds, You get a polypeptide, which is just a line of amino acids. And then that polypeptide will fold. When it folds, you have your protein. 12, what type of bond is formed between amino acids? Peptide bond. That's why it's called a polypeptide at the end. And then protein synthesis flow chart. I'm just going to go ahead and read through it. The first part of protein synthesis is transcription. And remember, transcription has that C. Translation has an L. C comes before L in the alphabet, so transcription comes first. Transcription takes place in the nucleus, where DNA is decoded onto mRNA. mRNA leaves through a nuclear pore and goes to a ribosome. The second part of protein synthesis is translation, where tRNA anticodons bond with mRNA codons. Then rRNA creates peptide bonds between amino acids, creating a protein. Okay?